Okay, so today I'm going to be talking about microorganism analysis with a qNano. First I'd like to say that the first three speakers set the bar quite high. I haven't cured cancer and I haven't published in Nature materials. Okay, this work was carried out by mainly me, Frankie Rawson and Samu from Izon. Frankie was with us as a postdoc at Canterbury. It was financially supported by Alison Downard and Keith Burrow and the Magdalene Institute. What I'm going to tell you about today is like a rather incomplete picture. It's ongoing research. Many times you might think, oh, why haven't they done this? Well, the answer to all these questions will be not yet. It'll be very soon. Okay, so now analyzing microorganisms by qNano, why and why. Counted bacteria, traditional techniques essentially is plating, well, more plating, and then there's some like newcomers like fluorescence-based assays. If you don't know about plating, there's a very cool bit such as this particular picture. This is like the geekiest yet coolest thing anyone's ever seen. This is all bacteria expressing GFP, brown FP, blue FP, these old fluorescent proteins. So these are all genetically modified bacteria. This is all bacteria. This is very cool, but this is the boring bit, plating. This is what people do to count bacteria. They dilute and dilute their samples until they can plate them on agar-agar, and then once you've reached a high enough dilution, you can actually count after a day of incubation. It's very long a day. You can count colonies, and you count them by eye. And you sit there on top of your plate and you count them one by one, and it takes forever. Okay, there's a bunch of techniques that are currently being developed, most of them involve fluorescence because it's super sensitive. However, these most often involve rather expensive pieces of kit. I know, for example, a French company that's trying to develop this a fluorescence-based assay, and they sell their piece of kit for 300,000 euros. So if you compare that to a $20,000 qNanos, a world of difference. Why do we count bacteria? Health and safety. Health and safety as well. And then there's protein expression in microbiology labs. Food and beer industry is very interested in this. And just general fundamental research microbiology. But it is very important. Okay, passing bacteria through a nanopore, what challenges are we facing? So the bacterial dimensions typically you're looking at between 1 and 10 microns. These are typical bacterial dimensions. You might encounter bacteria that are smaller, maybe 600, 700 nanometers, but mainly it falls into that range. The shapes are very variable. I've selected 4 pictures. 3 of them are mine actually. This one, and this one. This one was taken by the most famous microscopist for bacteria up in Hawaii. So you might encounter things like Fusobacterium, which is a very long rod-shaped bacteria. The dimensions are typically 10 microns in length and 1 micron across. This is a co-culture of E. coli and Staphylococcus aureus. E. coli is a short rod-shaped bacteria, and Staphylococcus aureus is a cocci, so a very round sphere. Then you get the funny ones such as V-shapes, and this one, which this is called Clostridium tetani. I don't know why, but the French call it the pregnant woman. I live at this too. In the higher orders, you encounter coccus, diplococcus, buzz, etc. So essentially cocci, such as Staphylococcus aureus, they tend to arrange in higher order arrangements. So you get these sort of grape shape, you get the strings of beads, all sorts. It's not always easy. Okay, this is insulting really, because I'm pretty sure you've all heard about the qNano and how it works. And if you haven't, okay, the best people in the world are here to tell you about it. Quick and dirty, does it work or not? Well, yes, it works, it's fantastic. I was not the first person to do this. Sam actually did it way before me. It was kind of straightforward for me to actually manage it. You don't need to try 1,000 different conditions. Basically, you just put your bacterial suspension on top of the nanopore, apply some pressure because we noticed that you can never get blockades without pressure, and you see like billions of blockades. So main observation is seeing these. In this particular case, I was looking at E. coli, which is genetically modified in the mouse. That doesn't really matter. It's a rod-shaped and gram-negative bacteria. Pore size is about 4 microns, and E. coli is generally regarded as measuring about 2 microns in length, with the standards that you get with a qNano. However, most of the blockades are of decent size and shape. We tried a few quick experiments, such as serial dilution. How does the system respond to serial dilution? So the first answer is, weirdly, you would expect the blockade rate for the nanopore to decrease linearly with the dilution factor. In my hands, it decreased linearly with the log. So my first question was I thinking like a first-year student? Answer is probably because I can't see any reason why we shouldn't get linear response. I had the pleasure to work with Frankie Rawson, who was a bit of everything. He was a jack of all trades, but his main expertise was in microbiology. And so together we did this toxicity study where we were looking at the effect of ethanol on bacterial count. So as you can imagine, this has huge implications in all sorts of fields. In this case, we were working with Proteus vulgaris. You find infections with Proteus vulgaris, nosocomial infections in the hospital. This is again a rod-shaped and gram-negative bacteria. So if you look at it initially, what you see is quite encouraging. This is increasing dose of ethanol as a model toxin. So the blockade rate is gradually decreasing, and this is exactly what I wanted to see. This is brilliant. Then we compared it to plating, the all-in-boring technique, and plating is shown here in yellow, and the qNano results are shown here in red. So if you want to be happy, you only look at the end, and you see that roughly the qNano and the plating technique gave us the same results, essentially, more or less. Then in the middle bit, it kind of agrees. And there's two main points of difference, at 0% ethanol and 25% ethanol. I honestly can't think of why this particular result was there, at 0% ethanol, but some things that might be interesting is, for example, at 25% ethanol, I personally think that we were able to pick up bacteria that was still living, but yet its vitality was diminished. So it wasn't going to later spread on the agar-agar when it was plated, but yet again we're able to see it with the qNano. So this has interesting implications because it's not because the bacteria is has a lower vitality that it's less toxic. If you take, for instance, something basic like E. coli, one of the main toxins is MPS, the lipopolysaccharide. It's not because the protein has low vitality that it's not toxic. So it's still interesting to see these bacteria, and that's quite a strong point that's making quite a strong case for qNano. And then this is actually the last experiment I'll be showing you today. Can we distinguish different microorganisms with the nanopore? In this experiment, we try to compare a protein that we've seen before, Proteus vulgaris, and just something basic, baker's yeast. Okay, Proteus vulgaris is shown here as with all the dots being blockades and their translocation duration, so the time that it takes the bacteria to go through the pore as a function of the particle diameter. And in the grey is what is shown is Saccharomyces cerevisiae, so baker's yeast. If you can see well enough, you might notice that these two populations are separated, and here shown as a graph, it's actually easier to see. As of today, I've never been able to, in a single run with a mixed population of these two microorganisms, distinguish two populations, but I'm quite confident that it should be feasible in the future. And if you take the case from baker's yeast, we got a very beautiful Gaussian shaped curve. This is actually all I wanted to show you today. The conclusion of this work was that qNano is indeed suitable for the study of bacteria and yeast, the findings, sometimes you might be puzzled or disappointed, but if we have enough time, we'll investigate these puzzling results, and I'm quite confident there's some interesting there. The microorganisms we've analyzed so far include two types of yeast, Archila adenivorans, which is relevant for fuel cell work, and Saccharomyces cerevisiae yeast. We've analyzed three different types of bacilli, so Proteus vulgaris, E. coli, and Bacillus subtilis. The cocci and Nextonale, they might prove more difficult because of their budding characteristics that I showed you before. And then there's a bunch of interesting things that might be worth trying on the qNano, such as parakeet, which are essentially bacteria with a spiral shape, and other things such as the bacteria that causes tetanus, Clostridium tetani, because it has a funny, just a pregnant woman shape. This is what I wanted to show you today. Thank you very much for your attention.